Professor Hoover, what is it that convinces you that life is not restricted to Earth?
I am absolutely convinced that life is not restricted to the planet Earth because I have found the remains of life forms that are absolutely, conclusively extraterrestrial. The Argelia meteorite that landed in France in 1864, the Murchison meteorite that landed in Australia in 1969, there is no question whatsoever in the entire scientific community that those are meteorites and they are extraterrestrial in nature. When I take those meteorites and I break them under very precise conditions in flame-sterilized pans with totally propane torch flame-sterilized tools and fracture the meteorite and put it on an electron microscopy stub with the freshly fractured surface up and then put it into the field emission scanning electron microscope, I find the obvious and recognizable remains of biological entities. And that has proven to me that since these remains are clearly embedded in freshly fractured interior pieces of the meteorite, that they are the remains of microorganisms and therefore they represent life.
So why then do skeptics or debunkers of such claims say that what you're looking at really are microorganisms as a result of contamination when the meteorites would hit the ground or hit the snow, and that somehow these microorganisms found their way onto or into the meteorite?
The only explanation is that they either did not read the papers I have published, or if they did read them, they simply did not understand them. And the reason that I can say that these are extraterrestrial is because with the scanning electron microscope that I use, it is possible to determine the relative abundance of the different elements that are present in the fossilized remains. And I can see elements from boron and above. And when I look at those samples that have the morphological characteristics of being cyanobacteria or other kinds of recognizable microbial entities, those that are interesting and exciting, I take the electron beam and focus it on a particular spot of the filament, and then the excited radiation that comes off, the X-ray radiation, is analyzed by the instrument in the electron microscope, and it tells me that it has this much carbon and this much oxygen and this much magnesium and sulfur and so on. But the point is, in these fossilized remains, it is almost never that you find detectable levels of nitrogen. And that is profoundly important because every living thing on Earth contains nitrogen. It has to have nitrogen for the amino acids, it has to have nitrogen for the proteins, it has to have nitrogen for every DNA and every RNA molecule in every cell. And after living organisms die, that nitrogen slowly goes away from the organism and goes back into the atmosphere as nitrogen gas. But that is a long process. It takes millions of years. These meteorites, Argelia landed in 1864, Murchison landed in 1969, Polonnaruwa on the 29th of December 2012. So these meteorites have not possibly been on Earth long enough for something to have crawled in after they landed and then the nitrogen to have disappeared. Anything that crawled into the Argelia meteorite after 1864 that was biological would have detectable nitrogen.
Is it frustrating to hear from essentially your peers who say that no, this, he's not right. We can't prove, or he can't prove, nobody can prove this. It almost sounds to me like, is it possible that some of these people, maybe all of these people, are afraid to acknowledge this?
I don't understand the reason, but in point of fact, these critics will not debate me in an open scientific forum. I would be perfectly willing to go to the Cosmos Club in Washington, to go to any university and have an academic debate and scientific discussion, and if they wish to say that these structures are recent contaminants, then I would challenge them to explain how these recent biological contaminants do not contain nitrogen. But it's even more important than that. You see, I'm not the only one that studies these meteorites. These meteorites have been studied by scientists ever since they landed. Lay in 1806, Arguello in 1864, Murchison. All three of these meteorites have been very extensively studied because Ci1 carbonaceous chondrites are extremely precious and extremely rare. They have been studied extensively by scientists, and when the scientists have studied them, they found all sorts of exciting things in them. Amino acids, very, very important, complex biological molecules. For example, they found nucleobases. They find uracil, they find adenine, they find guanine. But they don't find thymine, and they don't find cytosine. Now, these 5 nucleobases are critical because they are essential in molecules of DNA and molecules of RNA, and we do not know of any life on Earth that can exist without DNA and RNA. Well, of course, viruses can get by without DNA, but all of the cells of bacteria and archaea and all the eukaryotic cells require both DNA and RNA. The problem is you cannot have DNA and RNA without having all 5 of those nucleobases. So if a cyanobacteria had crawled into my meteorite in 1864, then when Sandra Pizzarella or John Carriage or Jeffrey Beta or any other scientists, Mike Engel, that study the amino acids and study the organics within those meteorites, they would have found all 5 of the nucleobases and all 20 protein amino acids if they were contaminated by modern biology. They couldn't possibly contaminate it with modern biology and leave out the life-critical biomolecules.
Why is there this resistance to acknowledging the work that you do? What's going on here?
Well, I think that someone like you should ask them that question. How do you explain this argument that he's made? Stephen Benner knows very well that you cannot contaminate a meteorite with modern biologicals without putting in all 20 protein amino acids and without putting in all 5 nucleobases. It is not possible scientifically. So the absence of those life-critical biomolecules in the carbonaceous meteorites proves that they are not contaminated. Now, I guess they might say, well, all of our samples of the Arguello meteorite and all of our samples of the Murchison meteorite that we've studied are not contaminated, but all of the samples of those meteorites that Richard Hoover has studied, all of those have been contaminated.
I see. How convenient.
Yes, I mean, they might come up with that kind of an explanation, but that's just because I'm just extra lucky, I guess.
Well, someone has to be. Well, okay, so now let's just take it off the Earth and off of the carbonaceous meteorites altogether. I've always been fascinated with Mars, and so, I've always thought, I've seen a lot of interesting pictures of Mars that say to me, there's something going on here that really needs to be looked at, and there's a combination of these old fossilized organisms versus more recently fossilized organisms or currently living. Do you think that the variety of missions that we've sent to Mars, all the various rovers who have gone around, I'm turning them into humans, all the missions that we've sent to Mars, have they come up with, do you think, evidence of not just previous water, but present-day water that could be supporting some kind of life?
There is absolutely no doubt that there is water on Mars today. And I mean frozen and liquid H2O. Now, there's all this excitement about, gee, we think we found evidence, read in the paper just a few days ago, that at one point in the ancient past, liquid water was flowing on Mars. To me, that was a very, very strange thing because, of course, the pictures that they were showing were obtained a few years ago, and of these streaks in the slopes of some of the craters. But the fascinating thing is that Viking, in 1976, found that the north polar cap of Mars was frozen H2O. Now, the problem is, H2O is water. It does not matter if it is frozen, which we call ice. But ice, when it is frozen H2O, it is still water. Gaseous H2O is still water. And liquid H2O is still water. Now, the reason that I consider that significant is while I was at NASA doing the astrobiology research there, I had a wonderful opportunity. I went to many of the hostile environments on Earth. I had 3 expeditions to Antarctica. I had expeditions to the Kvarkivik Glacier of the Vatnajökull ice cap of Iceland. I had several expeditions to the Matanuska Glacier of Alaska. And I spent, and the Fox Tunnel of Alaska, and I spent a lot of time chopping out samples of ice, and every sample of ice that I extracted I could put on a microscope slide and put a cover slip on top of it, and as the ice melted, you would see bacteria come out and start swimming. There are, and this was shown on movies that were made, films that were made by the BBC and the Discovery Channel, and so these films have been produced and are readily available, where they saw the actual process of the ice being taken, put on sterile microscope slide, and out come the bacteria and they start swimming. Now, why is that possible? Well, there have been a number of scientists that have shown that many bacteria have ice-active substances, enzymes in the exterior of the cellular coating of the bacterium, and the same way with many diatoms. And what this does is it changes the melting point of ice. And so if you have one of these bacteria in a frozen glacier, the exterior surface can cause the ice to become liquid, and so that organism is now no longer in a frozen sheet of ice, but is in an ocean, as it were. And if it has the ability to swim forward and melt more ice and allow other ice to freeze behind it, as it goes into fresh ice, the nutrients that are trapped within the ice come out, the minerals and so forth, and the bacterium can use them to carry out its organotrophic metabolism or its photosynthesis using sunlight and utilizing the materials that it needs to make new cells. And therefore, it is absolutely certain that life exists in ice. Well, the polar cap of Mars and the permafrost of Mars contains ice. And also, when the sun shines on dark rocks on the surface of Mars, they heat up. And in fact, when Viking was sitting there on the surface of Mars, on some summer days, the surface got up to 60 degrees Fahrenheit. Now, 60 degrees Fahrenheit melts ice in Huntsville. 60 degrees Fahrenheit will melt ice on Mars. And therefore, any ice that is right below the surface of the regolith would be melted.
Now, does that have anything to do with apparently an amazing image that you talk about taken by the rover Opportunity in 2004, that you would describe what that image is, because we'll be showing that.
I just became aware of this about a year ago. But Opportunity rover in 2004 took an image of a fascinating structure on Mars that shows structural features that are consistent with organisms on Earth known as crinoids. And a crinoid is an echinoderm. It's a very, very complicated organism. It's not a bacterium. It's not a cyanobacteria. It's not a diatom, even though diatoms are eukaryotes. Crinoids are echinoderms, like starfish. Starfish are echinoderms. We have living crinoids on Earth today. They go back far in the fossil record. Okay. And if you actually go on YouTube, you can see beautiful pictures where starfish are chasing a crinoid and it's running across the ocean floor to try to escape from being eaten. So these are animals. These are animals.
And you say they're on Mars?
And I'm saying that Opportunity took a photograph on Mars that shows features that are consistent with what we know of terrestrial crinoids, with pentagonal plates, with radial plates, with arms that come up away from the plates and actually branch. These are segmented arms, and they show branching, which is called bifurcation. And I've spent a lot of time collecting fossils, and I've collected a lot of crinoid crowns. This is the head of the crinoid as opposed to the stem.
Okay.
And when I looked at that, I immediately recognized it as a crinoid. I showed it to colleagues and friends. I have a friend that has written a book on crinoids, and he took one look at it and immediately knew that this was a crinoid. Now, the fascinating thing is, here you have a possible fossil of a very interesting organism in a rock on Mars, and 3.5 hours after that photograph was done, that rock was destroyed by the rock abrasion tool, at least the area where the fossil was located. I should say possible fossil.
The rock was destroyed?
The area where the possible fossil was located was ground to dust using the rock abrasion tool.
Oh, I see. In other words, humans were responsible for the destruction of the fossil.
Obviously, a decision was made in some way to utilize the mechanism on the rover to grind away the surface of that object.
And destroy the evidence.
And destroy the structure. Now, I once asked David McKay about this, David McKay, who did the studies of the Allen Hills meteorite, and he explained that that was done to look at the inside, that he knew all about it, and that it was done to look at the inside, looking for carbon or something. Right. Well, the problem is, anybody that does much in the field of paleontology knows that you don't have to find carbon to find a fossil. In the Huntsville area, we have lots of beautifully agatized blastoids called pyramites. And those have been converted to agate. They're silicified. And if I were to break one open and look for carbon in it, I wouldn't find any. But I know absolutely for a fact that it was a fossilized remain of a once living organism.
Who's making these kinds of decisions to destroy this kind of evidence, and why do they not want us to know about this?
Well, when you say I might not know the answer is absolutely certain I have no idea who made that decision. Yeah. This happened in 2004. By the way, the photographs are available on the web, and I can give you the frame numbers, and you can locate them on the web to see that what I'm saying is correct. Yeah. I believe those photographs that are on the web are authentic, legitimate pictures of that particular area. I believe they're authentic, legitimate information with regard to what happened to that possible fossil. And if a paleontologist finds on Earth a rock containing an interesting fossil, they collect it. You would never have a paleontologist say, gee, that may represent a new genus of life on Earth. Where's my rock hammer? I want to smash that to bits.
It brings us all into the idea of, well, conspiracy theories. Is there conspiracy theory, and what's it for? Why don't they want us to know? It can get totally crazy, but it's not like you don't know what you're talking about. You know, I've read your bio. you're like a heavyweight in science and in NASA, 40 plus years.
46 years in NASA before I retired.
And I know that in the 1990s, NASA awarded you like the inventor of the year. Yes. And so you're someone who knows what's going on. And so when you start going public on these things, between this image on Mars or what's happening inside some carbonaceous meteorites, people are trying to stop you from different angles. How do you feel about that? What do you think is going on there?
The first paper I published about microfossils in meteorites was in 1997, and it was in a conference that I chaired in San Diego called Instruments, Methods, and Missions for the Investigation of Extraterrestrial Microorganisms.
Did you come up with that title?
I did.
Okay.
And the book is available on the web. And at that meeting, which I organized, we had brilliant and very distinguished scientists there. David McKay came and talked about the Allen Hills meteorite results. We had Tommy Gold, of the Hoyle-Bondi-Gold theory of steady-state theory of the origin of the universe, and Tommy Gold, the one that predicted the deep hot biosphere. We had John Carriage, a famous meteoriticist, Jeffrey Beta, a famous meteoriticist. There were Tullis Onstadt, who had been doing research on the deep hot biosphere, the microorganisms deep in the South African gold mines. And we had 2 star panels which was filmed and then broadcast on television. So the point, and I did a paper and presented and published a paper on microfossils in meteorites at that meeting, and at every subsequent meeting I have given updates and published new data and new information. And then I published a peer-reviewed paper in the volume Perspectives in Astrobiology, which, by the way, was a NATO Advanced Study Institute that we held in Hanya, Crete, and it was co-sponsored by the National Aeronautics and Space Administration. And the paper in that volume also described microfossils in meteorites, and it was not only peer-reviewed, all of these papers went through the NASA approval process and the clearance process, so NASA was fully aware of all of them. So the point is for people to say, oh, gee, we didn't know, we didn't know. It makes no sense.
I want to know your opinion, because here we are at the International UFO Congress. What's your opinion on UFOs? And I don't just mean all things that people think that there are spaceships from Mars or whatever, the truly unexplained objects that really have not been given any possible explanation, and they're very bizarre, a lot of credible eyewitnesses. What do you think?
I know for a fact that there are a lot of credible eyewitnesses. I frankly have to tell you, I do not know what is going on with regard to the subject of UFOs. I do know that there have been events that have happened that are very, very difficult to explain. And one hypothesis is that these are craft from somewhere else. But what I have problem with is we live in an era of lots of cameras, and there are cameras everywhere. And I would like to see not only good high-resolution videos of these, but I would really like for somebody to bring a piece of metal or a fountain pen or an iPad from an extraterrestrial that, you know, a computer chip, something that you could take in your hands and say, look, we have analyzed this, and this comes from element number 127 that has not yet been developed, not yet been found anywhere on the planet Earth. So, I mean, that's the kind of hard evidence that would really make it possible to say, yea, definitively, we do know that these are extraterrestrial craft. Right now, I find it fascinating that there is continuing to be a large body of information and basically the kind of eyewitness testimony. But of course the problem is with eyewitness testimony is if I don't see it myself, I have trouble believing it when someone else tells me, if it's something that is very, very astonishing.